Okay.
Sounds good. Okay.
Alright. Yep.
Yeah. Okay. So, what is biodiversity and why is it important?
Well, first off, what it is. What biodiversity is really is a collective term for what we used to call biological diversity, and it's what you used to call before that all the species in a place and what they do. So it was a long sentence, and that's all collapsed down into the word biodiversity. So it's everything that's there and all the things they do with each other when they're there. Now the second question is, why should we care? There's two basic answers as to why we should care. One basic answer is that we, you, me, all of us, are the product of, more recently, 6 million years and perhaps 100 million years before that, of selective pressures from that biodiversity to make you what you are. That biodiversity gave you color vision, gave you the ability to smell, gave you your very complicated brain, gave you sound and the ability to appreciate and use sound. It gave you the reason to have words. It gave you all the different things we think of as ordinary human traits are the consequence of natural selection by that wild world out there. Now, of course, there's been natural selection by other humans as well, but they were also created by that wild world out there. So that's the first side of the answer, is that we are in fact a product of it. So if you want to throw away the mold and pretend that, oh, we just, fell out of the sky somewhere, well, you can do that, but I don't come from that world. I believe that it's very useful to understand what shaped me, what made me what I am, and it helps me understand why I react the way I do. The other side of it is the practical right now reason, and that is that all of us, all the time, in diseases, in food, in music, in sound, in smell, all these different things are in fact interacting with biodiversity. Every time you eat a meal, you're consuming biodiversity. Every time you get sick, you're being parasitized by biodiversity. Every time you hear complicated sounds out there, biodiversity is generating those sounds, whether they come from a bird or they come from the rain or whether they come from a windstorm, I don't care what it is. So that if you, in fact, keep the pieces that are doing these things there, you will lead a much, much richer life as a human being. And you'll be able to understand a lot more. Instead of saying, oh, I have a stomachache and God must hate me. You understand that you have a stomachache because there's some bad guy inside of you or because you ate the wrong thing. It's all a matter of understanding more about our world. That's to me why we save biodiversity. Because if you wipe it off the map, you're then living in a white box.
So, can you tell me how genomics has been used to learn about and conserve biodiversity?
Well, for me, genomics come very late. I'm old enough so that I come from the pre-DNA days of understanding. For me, genetics became things like what happens when you cross a white guinea pig with a black guinea pig and you get spotted guinea pigs, right? That was genetics in my world. And it was that way right up until 2003. So I didn't become involved in what you're interviewing me for until the year 2003. Now, in the year 2003, I heard a university professor named Paul Hébert from the University of Guelph in Canada stand up in front of an audience and say, well, it's interesting to understand the whole genome and to get the whole genome, because in 2003 that was very much the target. But I'm interested in something else. I want to use a little piece of the genome to identify things. And the world of genome people at that time reacted very negatively to this. They did not want to hear a new kid on the block. They did not want to have competition in the granting agencies. They did not want to have competition for fame. And they shouted him down. But as I listened to him talk, I realized I was hearing what I read about in science fiction when I was in high school. You land on a funny planet, you know nothing. You take your thing out of your back pocket and you point it at things, and it says dangerous, not dangerous, edible, not edible, useful, not useful. And this was before Star Trek. I'm pre-television, all right? I don't even own a television set. And I heard him say this, and I realized, wow, he's talking about doing in his laboratory what those guys were talking about in their hand. So I stood up in the middle of this conference, me who knows nothing about genetics, and pulled my comb out of my back pocket. And I said, I want to see what you're talking about in something that costs what this costs. And 8 billion people on the planet can have it in their back pocket. So he turned around to me and said, you've raised the bar. And I said, yeah, but that's the only bar that counts. Because what he had in mind still was that you send the sample to him. He gets the genetic information in his laboratory at the University of Guelph, and he sends you back the results. I wanted to be able to do that on the spot, in real time, now, repeatedly, for anyone. The school child, the farmer, the game warden, the doctor in the hospital, I don't care who you are. So I challenged him. I said, if I send you a bunch of butterflies that I've reared, so I know what they are, and nobody else knows what they are, can you tell me which ones are different species? He said, well, okay. And so I sent him a box of butterfly. It had 18 butterflies in it. And a week later, I got an email back. And he told me exactly which ones were males and which were females to go together, which ones were different, which ones were the same. And I can say that I was the only person on the planet who could do that before that, because I did it by finding the caterpillars and rearing the caterpillars, which is what I do as a biologist. I find caterpillars and I rear caterpillars. So I was very impressed. So I called him back and said, look, I have a huge collection of voucher specimens, tens of thousands of them. Would you like them to be your guinea pig? Because all he needed was one leg of each specimen to get the information he wanted. And he said, yes. So in 2003, we started this together, working together. Now, having said that, I then had to go and get an undergraduate biology book and read about mitochondria and read about genetics and teach me myself all the things that I should have known 20 years ago, 30 years ago, all right? But I came quickly to understand what we're really talking about. From my standpoint, genomics is going into the genome of anything, a rabbit, you, a lettuce plant, I don't care who it is, and taking out a little 650 base pair segment that we get the same segment from each of these different species. And we discover that that little 650 base pair segment is a little bit different from each species to the next, to the next, to the next, to the next. So if you have that little piece, that one little word, it's like having a collection of all the license plates on all the cars in the world. So all you got to do is give me the license plate and I can say, that's a, and now we know what it is, because I look it up in the registry where the license plates are registered. So really what I'm after is the license plate that goes with the species. Now, Paul elected to use the word DNA barcode for that, analogizing it with the barcode on a can of tuna fish in the supermarket, which is a fine analogy, and people capture that analogy quite well. So at that point then, my life changed because two things happened. One is I realized that my goal should be to build a library of these little identification pieces. That required getting one verifiable sample from each species into Paul's hands so he could take out the 650 base pair piece. I don't do that. My job is to get the specimen to him, and then I get the result back. But Paul, being an academic like I am, puts all his data on the web, just like I put all my data on the web for public access. So he not only gets the sequence, but it also goes on a website called BOLD, where anybody in the world can take their sequence, stick it in, and ask, do you already have this sequence in your library? And if you do, what's the name you have for it? So it's an identification tool. So that's how we're using genomics. So once you understand that, you don't have to understand anything in a big complicated genomics laboratory. You just have to get the little piece of your bug and somehow get that little piece to him. And right now we use FedEx and DHL. I mean, you know, and to me it's just like sending your film in to Kodak in the old days. You sent your film in, they developed it, and you got the slides back, right, or the pictures back. You had no idea how Kodak did it, right? And that's where I am. Now, actually, as an academic, I'm curious, and so I try to understand how they do it. But I don't really have to understand that. Now, that's what I do, is I build the libraries. The second half is somebody has to build the gadget. Because what you really want is something that costs what this costs, nothing, so that every school child can have this, so that every chef in every restaurant can have this, so every mother who's got a sick kid can have this. And of course that changes when they do. That will change our relationship to biodiversity totally. Because right now, you and everybody else is basically what I call bio-illiterate, okay? The analogy again is, you were taught how to read in second grade, so you're literate, and so you use that for everything. But nobody taught you how to walk up to an ant on the pavement and know who the ant is, or when you eat some little green thing, what is it? Or you bite into a McDonald's hamburger, is it really a cow? Or is it a horse? Or is it chicken? Or is it a pig? All mixed together? Well, you have no way of doing that. We can't teach you that when you're in second grade. But if I give you this when you're in second grade, then you can read the biodiversity that's around you, okay? That's what we're talking about. Now, so there are gadgeteers out there actually making and inventing this. It's going through various evolutionary steps. Right now it's still, you're talking about several $100s for something that can do something like that. And it's not easily reusable, and it requires reagents and things like that. But it's all headed towards the simple, cheap, throwaway, reusable gadget, all right? That will change our whole world. Now, to try to put that in some kind of real-world context, let me show you a few boxes of butterflies I just brought back from Costa Rica, okay? These are big things. Okay, these are big butterflies and moths. They're like a bird. So if you want to identify a bird, what do? You get a field guide to the birds, right? You don't have to shoot it. You can see it, and it's blue and white, and it looks, and you look up page X in the field guide. You can do the same thing with big things like this. However, when you do that, what you discover is some of these big things that everybody knows the name of are in fact 2 or 3 or 4 things. Because when you look at their DNA barcode, you discover that, oops, there's 3 kinds of barcodes. And then you look back at the biology that goes behind each one of these. What did the caterpillar eat? Where does it live? How does it behave? And you discover each barcode goes with a different caterpillar who eats different things. But the adult looks the same. So the butterfly looks the same as it did before, okay? But these are the great big things that everybody say, oh, I don't need barcodes to identify those things. In fact, you don't, but you do need to know that there are 5 different things out there in nature who look like this moth. And if I showed you all 5 of them, oh, they're all the same. But in fact, when you do the barcodes, you discover that they're not all the same, okay? So it not only allows you to identify, but to discover stuff that nobody thought was even there, all right? My easy analogy for that is, you know what orcas are, these black and white whales that eat, you know, in the ocean, SeaWorld and all of that. In fact, there are 3 kinds of orcas. They all have one name right now, but there are 3 species, and you can tell them apart by their barcodes. One of them eats fish, one of them eats small mammals, another one eats big mammals, which is where the name killer whale came from. Okay. So the point is simply that for big things like this, barcodes are not necessarily essential. You can use your visual for that, but they help you discover that this was in fact more than one thing. All right? Now that's for the big things. Now when we move down into medium-sized things like this, what happens is that the medium-sized things are a lot harder to tell apart, and there are a lot more of them who look very similar, even if you're looking very closely. So those things, barcoding becomes a lot more important in identifying these things. And with these, we discover the same thing I showed you with the big ones, and that is that with these little guys, they're also, you think, we know what that one is. It's the one with the orange stripe down the wing, right? And you discover the one with the orange stripe down the wing is in fact 5 different things. Okay. And so that's this size down here. Then we get into the really little things. And for these, the barcode is absolutely essential. Okay, we get into really, really, really small things. And under the microscope, yeah, you can often see differences, but who carries a microscope around in the field with you, right? Now, I said earlier that we are building the library of those DNA barcodes. Every one of these moths and butterflies that I've showed you, then we start back up and say, all insects have 6 legs. All mine have 5. Because every one of these has lost 1 leg. And that 1 leg is gone to Guelph. And I then, 2 weeks later, a month later, I get the DNA barcode back. The specimen itself is what we call a voucher specimen. That ends up in the Smithsonian Institution. So it's permanent storage. So in 10 years or 50 years, somebody's in an argument about, did that barcode really go with that specimen? Then you can go back and get this physical object, which is sitting there in permanent storage, and say, yes, or no, oops, there's an administrative error, there's some kind of data error in the background data or something. So that these have all lost 1 leg. These will all end up in the Smithsonian Institution. And in just a box full like that from Costa Rica, half of them don't even have names yet. They've not even been described by science. That will all come along. But now that DNA barcode becomes part of the description when you name it. So for every insect to have a name, every species to have a name, there's a formal legal process of describing it in a journal with photographs and description. Now we put the barcode in that description also. So you have what it physically looks like, and then you have this little piece of the genome. That then brings you to another bigger picture, which takes you back to the whole genome. Because now that you have the little piece, the genetics world can look across all those little pieces and say, wait a minute, where did that come from? So what they want to do is go back to this moth now and get the whole genome, because the little piece was a sort of a red flag that said there's something odd about this genome. And so now we can survey it. So if you build a library of tens of thousands, and that's what we're doing, of tens and thousands of species, the genome, the whole genome people who want to look at the whole thing can now scan across that library and look for little pieces that signal, oops, something interesting here, something more puzzling, something worthwhile spending the money and the time and the energy of getting a whole genome or some big piece of it out of there as well. And I suppose the last thing to mention about DNA barcodes is to emphasize that we use 1 piece, one 650 base pair piece for animals. For plants, we use 2 350 base pair pieces. So we're using a different piece of the genome for plants. And that's become that is because for magic reasons nobody really understands, and this is for the whole genome people to figure out. For plants, the piece we use for animals is basically the same from species to species to species to species. So it's useless. It doesn't have it. For me, it doesn't have any use. I can't use it for identification. It's like all the cars are the same license plate number. What good does that do? So you look for something else. And the something else turns out to be two shorter pieces, 350 base pairs, which can be used then to tell different plants apart. So if you go back to our gadget in the pocket someday, it would sort of have a perhaps two holes in it or two switches, and if it's plant part, it goes in one hole, and if it's an animal part, it goes in the other hole. That would be the way of dealing with that, or a possible way of dealing with that. Another thing to mention with respect to that is that the fungi, mushrooms and all of those guys, are in fact more closely related to you and me than they are to plants. And they may turn out to be best done with yet a third barcode, okay? Because they're neither animal nor plant, really. They're kind of another whole group. And so that's something that needs to be worked out. And I suppose the last thing to mention is that as getting the whole genome out gets cheaper and cheaper and quicker, it may be to the point where someday we don't even bother to get the little piece. We just take the whole genome. That, though, would mean enormous bulky quantities of data that needs to be analyzed. Where the neat thing about this is this is like running license plates on cars. You know, you're putting in a 6-digit number and the highway patrolman says immediately, I know what that car is. So there are balances between cost and efficiency. And the other side of it is that, of course, the people who are willing to go to the time and effort to get all these specimens and build a library, going out now and getting all those specimens to do whole genomes, be a whole nother package of work, and we're dealing with millions and millions of species. Probably on the Earth's surface there's between 20 and 30 million species of things. That's an awful lot of genomes. Okay. Now, where I guess the last thing to mention that's happening is right now, to get the 650 base pair piece out of the genome, we go through a complicated process called PCR, which will show up somewhere in other interviews. It takes machinery, it takes time, it takes reagents and all of that. Well, one way to get down to having the cheap barcoder is to try to collapse the PCR process into very small things. My own opinion and guesswork from watching this process is that that's probably a dead end. In other words, you can only reduce it down so much and you have to change gears. And where the gears are changing is going to probably direct read of the DNA strand, which involves basically pulling it through a hole, and there are sensors in the margins of the hole which tell you which base pair it is as it goes through. So it's like reading the beads on a necklace, you know. So there the trick is just getting that DNA thing stuck in that hole and pulling it through and having it read like that. So PCR disappears. It disappears from the system. The other neat thing that's emerging is that right now we're thinking in terms of you or the school child or the farmer who picks up one leg off one insect and sticks it in a hole. We now realize that you can take a big sample out of the ocean, out of a river, out of wherever, and barcode the whole sample at one time. And we're in the process right now of testing that by, they say to me, Dan, I need one leg from 1,000 different species in one tube, which they then grind up and make soup out of that. So they're all mixed together. And then they barcode the whole sample. And they come back to me and say, all right, here's the list of what we think was in that sample. And they're right now at 80% to 90% correct. So we're headed in that direction. And there are statements already existing like, if you give me a liter of water out of a river in Europe, I can tell you all the species of frogs that live in that river, in the whole river system, just from a sample of water. So there are many advances being made, but I want to emphasize here, it's the use of genomics. It's not exploring genomics, really. It's very practical engineering kinds of use. And we need that along with the esoteric exploring part as well. Okay.
So one follow-up question. When do you think this pocket scanner, when do you think the pocket scanner will be available at?
Well,
sort of masses?
I'm asked that question. I'm asked the question frequently as to when will a gadget be available. My own prediction from watching the way the economic and the world works is within 5 years. It might cost $2 by then. It might not be the cost of a couple of pennies, but what I see is within 5 years there will be a personal pocket scanner that you can have. Then the question will be, how fast can we build a library against which you do your referencing? What I see happening is that you're standing in southern Venezuela, and a caterpillar just stings your hand, and you want to know, should I worry about that? Or it's just pain, and who cares, you know? And so you kill the caterpillar, or you get a piece of him, and you get it on your thing, and it goes through a wireless world, it goes to a central system. And the system says back to you, we don't have that barcode. Where are you? And you push a button on your iPhone or whatever you have that's the equivalent of that, and it registers then where that barcode came from, and then it says to you, and what does it look like? So you take your iPhone camera and you go click and you take a photograph of it, and then that's in the thing. So now they have a photograph, they have the barcode, they have a locality. From the photograph, it's probably possible to say, that was a caterpillar, that wasn't a snail, that wasn't a mouse. And so I can predict that there'll be a system of taxonomists out there who effectively be people who look at that photograph and say, oh, that's another species of Hylesia in the Saturniidae, and so now we have a Latin name that goes with it. It might not be a described species of Hylesia, but at least it's in that genus Hylesia. And then it will say to you, are you willing to collect it for us? And often you would say, no, I'm a tourist, I don't have time to do that. But if you're a scientist or somebody who really cares, an agricultural worker or whatever, then you might collect it and send it to some central place. So you gradually build the library by users. And if you've got 8 billion people all doing that, you can see where the library could grow very quickly, especially around the things that people come in contact with, the things that matter. You eat them, they bite you, that sort of stuff. I would expect those libraries to build quite quickly. There'll be all kinds of esoteric things that nobody's ever heard of and will never ever see, which make a longer time for the library to build. But right now, the resource in short supply is actually the gadget. But once the gadget's in place, then I see the resource in short supply is the building the barcode library and having a community of scientists or whatever practitioners who are effectively serving the function of providing the taxonomy that goes with these things that now suddenly we can read. So suddenly it's just like teaching you to read, and you say, wow, I want to read more books. And somebody says, sorry, the library's closed. And you say, but why is the library closed? Because we don't have any call numbers on the books, so we can't find anything. All right? So it's like you go to the Library of Congress and they say, sorry, if you can't read Chinese, you can't go to the Library of Congress. Those are the blockages will appear of those natures. But right now the blockage is just getting a gadget built and building libraries as fast as we can to demonstrate to the world that it works. And in the scientific level, hey, it works, no problem. Yeah. Okay.
Good.